We're going to try out a CRISPR lab to see if we can turn green fluorescent protein, the gene encoding green fluorescent protein, if we can fix it in these cells that have a broken copy, if we can fix it using CRISPR-Cas9 and see production of green fluorescent protein in the cells in a few days. So the first thing we have to do is synchronize the cells so that they're all at the same stage of the cell cycle. So CRISPR makes double-stranded breaks in DNA, and that gives you the opportunity to insert sequence or take sequence out, to correct sequence or get rid of sequence you don't want. So those double-stranded breaks get corrected by a process called non-homologous end joining, and that happens in S phase, G1 phase, and G2 phase. So we're going to synchronize these cells at the end of S before the beginning of G1 so that they'll all be ready when we put the CRISPR components in for the double-stranded breaks to occur and then to do the correction, to insert the sequence that we want. So we're going to put the cells in some media. So I'm going to put 5 milliliters of media per 10 centimeter dish. These are tissue culture treated dishes. Trying to spread it over the surface of the dish so that it's even. A little bit. There we go. More or less. Okay, so now I'm going to add a bit of choline to the appropriate concentration to each dish. Okay, now we're going to add cells. These guys are at 2.5 million per 100 microliters. So we're going to put 100 microliters per dish. Okay, so then we just tip the dishes to get the cells evenly spaced. You don't want to swirl like this because that pushes them to the outside. So you want to rock back and forth in a couple directions. Then I'm going to put these in the incubator. They're going to go for 24 hours. By then the cells should be synchronized, and then we can do the rest of the CRISPR experiment. Okay, so the cells on this dish have been in the presence of a bit of choline for almost 24 hours, so they should all be arrested at the G1S interface. So now, before we try to do the CRISPR reaction, we want to release them from this block so that they can start cycling again and be in the right place to do the non-homologous end joining repair after CRISPR makes the double-stranded DNA cut. So I've checked them under the microscope. I wish I had a setup to show you what they look like, but they look fine. They're attached to the dish. So right now I'm going to remove the media that's on here that contains a bit of choline. I'm going to wash the plate twice with PBS, and then put complete media, so that's media that contains fetal calf serum and L-glutamine on these guys. and then they're going to sit for 4 hours so that they can start cycling again before we continue with the CRISPR reaction. And the phytocholine needs to be properly disposed of, so I'm going to put all the media and washes into this tube for proper disposal. Alright, so we're going to remove media. Okay, so I've removed the media from both dishes, and I'm going to wash them with 5 mLs of PBS per dish. These washes are just to ensure that we remove all the phytocholine so that the cells will go back into cycle. So I'm going to do that one more time. We really want to wash them well. Okay, now I'm going to put 5 mL of complete media on each plate, and we're going to put them back in the incubator for 4 hours. In this little tube, we have our 2 RNAs complex. So we now have to mix those with a little bit of a special type of media, as well as with the Cas9 nuclease to make the total complex that we're going to add to the cells. So I'm going to do that right now. I'm going to mix up this mixture, and then we're going to add that to cells, and then we're going to add the DNA that provides the template for changing the nucleotide that will allow GFP to be transcribed and translated. We're going to mix that all together in a cuvette, and then we're going to zap the cells to poke holes in their membrane so that this whole complex of DNA, single-stranded DNA, and RNA and Cas9 can all get into, hopefully, lots of the cells. We'll see on Friday. All right, so make these mixtures. And I'm going to make 2 mixtures because I want to do this twice. Just because I have enough reagents, and why not? Alright, so these tiny, tiny tubes. Alright, so it's 3.3 microliters of our special media that's called OptiMem per tube. And then 1.67 microliters of our Cas9 nuclease. nuclease. .78 microliters of this special buffer, which is here, special RNA buffer. And 2.2 microliters of that RNA that we complexed before. Okay. Alright, so we've got that. We've got our cells at the right concentration. And we've got our single-stranded DNA that's going to help us repair the mutation that keeps GFP from being transcribed and translated properly. Okay, so we're going to use these cuvettes. These are cuvettes. There's metal on each side. We're going to put cells in there. We're going to put the RNA Cas9 complex in there, and we're going to put our single-stranded DNA template in there. And then we're going to use a machine to apply electricity to the cells and poke holes in them so that our DNA and RNA Cas9 can get into the cells. We hope. All right, so I'm going to do this twice. So I'm going to put the cells in first because they're the biggest volume, and then I know I can mix the other stuff into the cells, the other reagents. All right, so cells. Here we go. And now 10 microliters of our CRISPR-Cas9 complex. All right, and then 1 tiny microliter of our DNA. Cap on this guy. Okay. Now, the protocol says to lightly flick the cuvette to mix. We want to make sure our cells and our CRISPR-Cas9 complex and our single-stranded DNA kind of repair template is all really well mixed together. Now for the moment of truth, sort of, one of the moments of truth. We've got our electroporator set the way that the protocol says, 250 volts, 2 pulses, each pulse 13 milliseconds with 1 second in between. So you can program the machine to do that. And then you put the cuvette into this little kind of cuvette holder that has metal on either side. So you make sure to get the metal sides of the cuvette against the metal there. And it's kind of set up in a way where it won't let you put it in the wrong way, which is nice. So I'm setting it in there, clicking that shut. And then the P is pulsing down here, so that tells me it's okay to press this pulse button. Okay, so we've electroporated our cells. And you can kind of tell that they're electroporated if you look at all the foaminess there on the top. The electricity kind of makes some bubbles and stuff. So that's one. The machine tells you if it did the pulse, but you can also tell by looking for that foaminess. So now, I'm going to take the contents of those cuvettes out, and I'm going to put them each in one well of a 6-well plate in 2 mils of complete media. And then in 3 days, we'll take a look at these under the microscope with the fluorescent scope and see if any of them are GFP positive. Fingers crossed. So, okay, so I'm going to label my plate. Complete media. Two mils per well. So I'm going to open these guys up. I'm going to go down in there and suck up all the media and cells. Put them in here. And then it's really hard to get everything, so I take a little bit of the media and I wash it around in the cuvette. So I want to try to get every bit of the cells that I can. Okay, so I'm going to do the same thing for this cuvette. So now they're in culture. I'm just going to take a peek at them before I put them in the incubator. And electroporation is pretty hard on cells. A bunch of them are going to die, but hopefully some of them will have holes poked in their membrane. The CRISPR-Cas9 and DNA will get in, repair the GFP mutation, and then those cells will start making GFP. So enough holes poked for the stuff to get in, but not too many so that the membrane can repair itself and the cells still live. Here's some of the cells from the transfection. We've got them on the EVOS microscope just under light, regular light. So you can see them here on the screen, sitting down on the plastic. There's a bunch of rounded up cells that are floating, and those cells are probably dead. The electroporation is not easy on the cells. It kills a certain number of them. But we've got some living ones here. So now if we switch to our green light channel, we'll be able to see if any of these cells are producing GFP. And you can see that not all of them are, but we've at least got a couple here that are quite bright for GFP. So our CRISPR experiment worked, yay! You never get 100% transfection of cells, especially with electroporation where so many cells die. But we've definitely got some green guys here, so that's really good. Yay! Okay, so here's a diagram of what we did in the CRISPR experiment. I think it's helpful to kind of see a diagram since you saw me manipulating a lot of things, but what was I really doing? So the first thing I did was to put the cells in culture with a chemical called acetylcholine that would synchronize them at the G1S interface. And then we released them from that synchronization so that the cells could start cycling again. And that was just to get them in the right, all the cells in the right phase of the cycle for this reaction to be able to take place. So then we made our RNA complex, our CRISPR RNA complex, which is made up of a spacer RNA and a scaffold RNA. So the spacer provides the specificity for where this RNA is going to bind to the DNA. And the combination of these two allow this to bind to the Cas9 enzyme. So the first thing we did was that reaction where we mixed these two together, and I put them at 95 degrees to melt all the RNA apart and then let it anneal back together. There's some complementary regions here, so this forms a complex. Then we mixed it with Cas9, and then we mixed all of this with cells. And the guide DNA that is going to, down here, sorry, that's going to repair the mutation in the GFP gene. So we mixed this complex, this CRISPR-Cas9 complex with cells. Sorry, I should put that up here, plus cells. And we added this DNA template with our single base pair change that will now make the GFP gene functional, and then we zapped it, we electroporated it with electricity, and then we put it back into culture. So then you have to give it a few days for this whole reaction to occur in the cells and for the cells to begin transcribing and translating GFP if this happens in the cells, if this repair function occurs. And as you saw in the data, not all the cells are bright green now 3 days after the experiment. Only those that were effectively transfected, didn't die from the electroporation, and had this reaction occur in them so that they could turn transcription and translation of the GFP gene back on. So we had success, yay, we saw green cells, and this is what was happening inside the cell.